So we will continue with the protein. We were discussing the conjugate protein. The conjugate protein contains the protein and non-protein part, which is called prosthetic group. Now, why the protein are able to conjugate with others? Because there are so many polar groups which are present in the amino acid, and they have a tendency to group with the other, you can say, other groups. Now we'll be discussing here some conjugate proteins such as chromoprotein is a type of conjugate protein which consists the non-protein or prosthetic part as pigment, and examples are hemocyanin, hemoglobin, cytochrome, flavoprotein, and all the pigment, you can say they are part of chromoprotein. Now, another is glycoprotein. The prosthetic part is carbohydrates such as heparin, immunoglobulins. Lipoprotein, the prosthetic part is lipids such as egg yolk. Phosphoprotein, the prosthetic part is phosphoric acid such as ovovitiline of egg, casinogen, phosphoproteins. Metalloprotein have the metal ion as a prosthetic part and such as carbonic anhydrase, even the hemoglobin which has iron as a prosthetic part, so it also falls into this category. Okay, now we are discussing the protein on the basis of the shape. They are divided into two categories, fibrous and globular. Let's discuss the fibrous protein. They are in the form of long and tough fiber, usually insoluble in water. They usually form structural component. Um, the examples are keratin, collagen, and fibrin. Keratin, as we have seen, is present on the outer layer of skin, nails, horn, feathers in animals. Collagen is found in white fibrous connective tissue in the muscles, dura mater of brain, a ground substance of cartilage and a bone. Fibrin occur in raw silk, a spider web, which have a great tensile strength. Now, globular protein, they are in globular shape, that is coil form, and the small globular proteins are soluble in water, whereas the large one are insoluble in water. Examples are albumin, globulin, gluten, enzymes, and hormone. Here, the gluten protein, which is present in the wheat, usually it is also present in some of the seed in the endosperm portion, and it is the storage product over there, so it is present in good amount in wheat, and it makes the dough elastic and offer favorable baking qualities. Now you should also know that sometimes some people have intolerance to gluten. That means they are unable to digest this wheat protein, or they may be allergic to this protein. Okay, so it is always advisable that along with the wheat you can consume certain other cereals like millet, corn, uh, and barley. And barley, it has less content of this gluten protein. So those who are intolerant to, you can say, gluten or wheat, because this is present in high amount in wheat, then they can switch on to those kind of uh, diet. Okay, now this is about the classification. Now we'll see the biological functions of protein. So I'll be discussing only type of protein. Structural, since they are the basic bodybuilding, what we call the protein or bodybuilding food. So if you see the structural protein, collagen, elastin, osin, which are present in muscle, osin in the bone, keratin, and microtubules, these are some of the structural protein, main structural protein. Then they also play a role in transport and carrier, as you have studied in cell, also studied in the muscular system. Enzyme we will do hemoglobin in the circulatory system, that how it transport the oxygen. Myoglobin, it transport the oxygen in muscles. Serum, albumin, all these are transport, act as a transport and carrier. Then there are some regulatory protein, as we have seen in case of hormones, some of them act as a receptor. Even in the cell membrane there are receptors. Then hormones, they do the regulate the different systems of the body, different metabolic pathways, and they exhibit buffer action because since they have cations and anions which can maintain the negative and positive charges in the cell and around the cell. So they maintain the pH, so hence they have the buffering action also. And then protective function, since the proteins are the one which forms the antibodies, they also form the blood clotting factors. Then catalytic function, because all the enzymes are protein. um, and storage function, like we have seen gluten which gets stored in the seed, casein, ovalbumin. So all these are the functions of protein. Now there is one important concept that I would like to discuss about the disulfide linkage. Cysteine is the amino acid, which contains the sulfur and which has sulfur hydryl group and can be easily oxidized. Oxidized means oxidation is either the addition of oxygen or removal of hydrogen. So it has a sulfur hydryl group. The hydrogen can easily be removed, so it can be easily oxidized to form a dimer containing disulfide bridge between two cysteine. Such dimer is known as a cysteine. So cysteine is converted to cysteine by forming the disulfide linkage. Cysteine containing region in enzymes, they forms the active site. Now another amino acid which contain the sulfur is methionine, and in methionine the sulfur group is attached to the methyl group. Thus the methionine is more hydrophobic, larger and less reactive than cysteine, so it will not form the disulfide linkage. So disulfide linkage is only formed by cysteine. The sulfur linkage stabilizes the 3D structure of protein, and the disulfide bond can be broken by addition of reducing agent such as beta mercaptoethanol and dithritol. Okay, so these are some detergent, some salts which can um, added, and they act as a reducing agent, and um, they can reduce the disulfide bond, and once they are reduced then 3D folding is lost and the stability of protein is destabilized. So if you look at here, these are cysteine, two molecules of cysteine. So when they are oxidized, that means their hydrogen is removed, the disulfide bond is created, and when it is reduced again it come back. So this structure is called cysteine, this is cysteine. So this is the basic difference between them. So you'll find that there are proteins which consist of cysteine bond, like our hair. Okay, they also carry the cysteine. Okay, so the curly hair has cysteine, and the straight hairs, the bond is oxidized, so it become the straight hair. So this is what is the basic principle which is used between the straightening of the hair. So this is all about the proteins in our syllabus. Thank you.